This is just a quick video going over some slides that I found of onion roots and whitefish, the two cells that you are going to be identifying and drawing and analyzing on your midterm. This is an onion root cell tissue sample, and you'll notice that the cells appear very square-ish, almost like a slice of cake, and generally we can really distinguish cells that are dividing or not dividing. If we start to see chromosomes concentrated sort of in the center, we can start to see genetic information being condensed, generally going to be prophase. Metaphase is going to be any point in which we see those chromosomes really getting lined up in the middle of the cell. Obviously, we can see cells that don't really have a lot of condensed visible DNA. We might see a stained center, which is the nucleus and the nucleolus, but we don't see the specific chromosomes. And then things like anaphase are going to be where we see those chromosomes being divided from the metaphase plate by the different centromeres. So this is a pretty good example of what you could expect to see on slides when you're looking through them in the microscopes in terms of cells that are in interphase, obviously not dividing, prophase, metaphase, anaphase. Obviously, if we started to see these two condense, we might be seeing cytokinesis. And note that there are some cells with very little visible stuff in it, and that might mean that that cell is not currently dividing. It might be in G0 state. It might also be dead because it's a tissue or a cell that hasn't been replaced. We can also see on a whitefish slide the different aspects of mitosis. So if we zoom in closely, here's a really good example of telophase. Some of you saw this last week where we have two cells without that cell membrane yet formed, so we can see how they're still conjoined, and we can see that the DNA has been uncondensed and the two nucleuses are forming. Prophase is going to be when we see that condensed chromosomes, not yet in individual sister chromatids yet, but it's very visible. This is a very faint metaphase, but we can see all of those chromosomes lined up. Another example right there. Here's anaphase. Anaphase sort of looks like two clusters of bananas being separated apart. So some good examples of that. And again, when we see cells that look like this, or this, or this, or this, or this, generally means that they are cells that are in G1 or G2. They're currently not dividing yet. Cells that are in G2 might have a bit more condensed chromosomes visible because they've been duplicated after S phase, but definitely these cells right here are all in sort of G1. I hope this helps. Please see me if you have any questions, and good luck this week.